I'm Seth Lerner. I'm a urologic oncologist. I work in Houston, Texas at Baylor College of Medicine. So this is a trial in progress. It's actually just getting started. It's a phase 1 trial. It's a novel drug delivery system developed by a company called Aura Biosciences. And it's a virus-like particle. So think about it as the protein shell of the virus, but without any of the nucleic acids. And it attaches specifically to bladder cancer cells, and it's coated on the surface with light-activated particles. So you use a laser of a very specified wavelength, and then it creates all these reactive oxygen particles that disrupt the cell membrane. So that's the method of cell kill. And then there's a secondary effect of these tumor-associated antigens, APCs, stimulating the immune system. So it's kind of a direct kill and then a stimulation of the immune system. So the trial is designed to actually work out some of those parameters. So it's a direct injection within the lamina propria around the base of the tumor. And we're doing this in both patients that are going to undergo a standard of care TURBT or cystectomy. It's what we call a window of opportunity trial. So using that window before surgery. And then we're also going to test both sort of intramural and intratumoral injection in the second phase. And one cohort of that will also get the laser light. So this is kind of working out the parameters and the safety of the drug delivery system and the drug itself. Well, I mean, the obvious thing is we're directly targeting the tumor itself. And then the hope is that by also stimulating the immune system, you may also come back and target, say, incipient CIS or dysplasia or other tumors within the bladder. Now, this is actually all comers. We wanted to try to simplify it because we're just really working out the technique and the safety. Very detailed pathologic examination to look at distribution of the agent, and then we'll figure out the specific target population. Yeah, it retains a very innovative approach to taking patients who are getting standard of care neoadjuvant chemotherapy, enriching that population by identifying patients who have alterations in DNA damage repair gene alterations, and if they have a complete clinical response, then observing them. And then if they don't have a gene alteration or they don't have a complete response, they go on to get definitive either radiation or surgery. They designed the trial, by the way, this is done at Fox Chase, Dan Geinesman, Betsy Plimak. It's really a very forward-thinking type of trial. They designed it with a non-inferiority margin of the lower bound of the confidence limits, I think, of 64%. Unfortunately, they didn't meet that. And so just in reading through the abstract that'll be presented later today, they're going to go back and kind of figure out the next steps. As far as bladder preservation goes, we're very excited about that. We're about, I say we, in the Southwest Oncology Group, a trial that we're doing in conjunction with NRG, Parminder Singh, Jason Istathu. We're about 3/4 of the way through the largest bladder-sparing trial done in the United States. We'll accrue about 475 patients. It's a randomization to standard of care chemoradiation therapy with or without atezolizumab. So, in a huge translational medicine program going on. I think the field is finally kind of beginning to embrace this from a clinical trial standpoint, and hopefully, and it's a standard of care in the NCCN guidelines as well. So.